today we have 2 more sessions, session 1 and session 2. Session 3 is the poster flash, and after that we have dinner and a social event. I'm going to give the heading, the chairing of the session to David Sprinchak with the chair of the next session, biology-oriented session.
Thanks, Nati. Okay, so we're continuing to the next session, biology aspect, and I'm excited to be a chair in that in this session. And the first speaker is Ronit Satchi Fainaro from medicine.
Okay, good afternoon, everybody, and thank you for inviting me to this really exciting day and conference. It's not really fair to put me after Peter Zieberger's talk. It was just fantastic. But I think we can all find some common grounds, and this is why we are all here. So I will start with a little bit of background on the chemical approach, how we approach things, and then, as Peter mentioned very nicely, we'll go deeper and deeper to the biology and never come out. So it all started from a fascinating set of provocative questions that the NIH and the NCI put in 2012. And one of them really intrigued us, which was what molecular or cellular events establish tumor dormancy after treatment and what leads to recurrence. I think this is a really important question, a really important clinical unmet need, as we all, in most of our papers, show very nicely a study of 15 to 30 days where we cured cancer in mice, but then nobody looks at what happens after 100 days, and this is the time where everything escapes and starts growing back, and this is actually recurrence. So we wanted to see what happens at that point that we think we have cured cancer, but actually we have some dormant tumors staying there at the size of 1 to 3 millimeter in size. So we went back to the literature and we found this really fascinating paper at the New England Journal of Medicine in '93, more than 20 years ago, where they did 16,000 autopsies in Sweden, and these were of people that died of trauma, of car accidents or suicides, nothing to do with diseases or definitely not cancer. And what they found was really striking. If you look here at the scheme and the numbers, about 46% of the men had these tiny tumors in cytocarcinomas in their prostate. About 39% of the women had these tiny in situ carcinomas. They were all avascular, about, again, 1 to 3 millimeters in size in their breasts, and virtually all, 99%, had these tiny lesions in their thyroid. That means that basically all of us have tiny in situ carcinomas in our body. They are asymptomatic. We don't know that they are there. We don't feel it. But if we look how many of those are clinically diagnosed, we're talking only about 1% for prostate and breast and 0.1% for thyroid carcinomas. So if we look at it way more optimistically, it means that we have something in our body that keeps those tumors dormant at the size of 1 millimeter without being able to recruit blood vessels, recruit the stroma, the fibroblasts, immune cells, to help them to progress to cancer, to progress to metastasis, and later on, even after treatment, we see some recurrence. So we wanted to know which manifestations are those of tumor dormancy, and we looked at it starts as the primary tumor, one of the early stages in tumor development, a stage in micrometastasis, and minimal residual disease following surgery or treatment. And if we look at it more schematically, it could be, let's take osteosarcoma, a bone tumor, which we recently published about it, a pair of dormant and fast-growing tumor in ACS Nano, where you can see that one of the early stages of primary tumor development, it can be following some circulating tumor cells that left the primary osteosarcoma and went to the lung and then sit there for a week, a month, even 20 or 30 years, and then grow as a big metastatic lesion. or minimal residual disease following treatment. It could be treatment or surgery where we are unable, or the surgeon is unable to see that there are some tumor cells left in the region. He closes up, and then we see the recurrence in the same site of the first primary tumor. So we looked at it a bit more philosophically, and we wanted to see how we characterize them. Are they, those cells, are they behaving as tourists? Do they arrive to that site and now start exploring the new area, the new country they arrived to? The lungs in the case of the osteosarcoma, brain in the case of melanoma or breast, or liver in the case of pancreatic cancer? Do they check what is around and start talking to the local inhabitants? Or are they more as an ambassador, looking and offering and telling everybody about the way, what happens in their own country? Meaning if I am a tumor cell coming from a bone neoplasm, like osteosarcoma, do I start making hydroxyapatite, the bone mineral? If I come from the breast, do I start having HER2 on the cells presenting those antigens, those receptors, when I am in the brain now? Do I start talking to those local stromal cells, meaning if we talk about the brain, these are the astrocytes, microglia, neurons, and others. So this is the ambassador type, but there is a third type. This is much more difficult to cure or to deal with, and this is the refugee, because they come under the radar. For breast, just to put it an analogy, this is the triple negative kind that we don't know how to cure. These have no identification. They do not want to be identified, where they come from, what have they done, and they don't want, they try very hard to be under the radar, so our immune system cannot detect them, and then they extravasate from the blood vessels, they sit in a foreign area, a foreign organ, and now they start proliferating slowly without anyone noticing them, and when we notice them, it's already too late because we are unable to treat them because they don't have any specific antigen. This is the reason I was asking about what are you targeting there. So if we go back to the ground, our basic questions are basically, what are the basic mechanisms that maintain the state of tumor dormancy? What are the events that initiate the transition from dormancy to aggressive disease, to have a crosstalk and interactions with the stroma, meaning the endothelial cells, those cells that create the blood vessels and that are recruited to the tumor in order to feed it with nutrients and oxygen, to the fibroblast, converting them to cancer-associated fibroblast, and to macrophages and the immune system, converting it from M1 to M2 macrophages. Is this process reversible? Can we re-educate those cells back to their form as they were when they were a dormant tumor? Not necessarily, this is not dormant cell, because we see that in those dormant tumors, they are metabolically active. They proliferate and they undergo apoptosis, so there is an equilibrium and they remain in that size as a dormant tissue, as a dormant tumor, but not as cellular dormancy. And then if we can define and understand the mechanisms by which cancer cells basically hijack the functions of normal stromal cells, and they take them as culprits, basically, can we design now, and this is the part of the chemistry in my lab, to design a new, in our case, is nanomedicines that disrupt those tumor host interactions. We would like to sabotage these interactions so they can be re-educated to either back to dormancy, like in those 16,000 people in Sweden, or completely eliminate and eradicate them. So the way we address these questions is first by creating models of dormant versus fast-growing angiogenic, meaning that this tumor already recruited blood vessels, recruited the microenvironment. Those pairs come from the same parental tissue of cancer. Then we characterize them by RNA sequencing, gene array, microRNA, proteomics, and we see, and to simplify things, let's say that we have here a protein that is overexpressed. Can we now manipulate it just by lentivirus, for example, silencing the gene in the dormant, and see, can we keep it as dormant now? And if then we have a validated target when we look at proliferation, migration, tumor progression, and then in the histology to see that we actually targeted and silenced that target, and by that inhibited tumor growth, then we move on to try and create an actual drug, meaning if we use an siRNA and we cannot use a lentivirus, can we use an siRNA in a nanoparticle, deliver it only to the site of the tumor, and then test it and evaluate it in vitro on tumor cells, then in vivo in mice, and hopefully move on to clinical setup. So this was the way we approached things until probably 3 years ago. Then we had some MDs, some neurosurgeons joining the lab, and we decided to reverse the whole process. So now we're starting from the surgery room. We take a chunk, the rest goes to the pathology. We take it to the lab, implant it in a mouse, and we have a PDX, a patient. We have the tumor inside, implanted in SCID mice. These are immunodeficient mice. And then we reverse the whole process and try it all the way around. So this is going very well for us, but it's very slow. It takes about 6 to 9 months to create a colony that is large enough of mice with the same tumor that we took from the patient in order to test our novel molecules and to see if we are able to inhibit that tumor, its growth and progression. And to come back mainly to patients that, as I mentioned, with glioblastoma, with brain tumors, or with pancreatic adenocarcinoma, that is the subject of my talk today, 9 months is a long time. It is no longer relevant to come to these patients with an answer after 9 months that we have found a compound that will inhibit their growth. And for that, we had to think a little bit out of the box and think how we can make things faster. Maybe now that I saw how fast your carbohydrate synthesizer is, we can go that way. But in the meanwhile, we're still going to the surgery room. But now before we're actually undergoing the surgery, the patient undergoes the surgery, we take its MRI or a CT scan. Now we do the image analysis and convert it to an STL file that a 3D printer knows how to deal with. And on the day of the surgery, we take again a chunk of the tumor, but now we dissociate the tumor to single cells, but this time it's not only tumor cells that we put on 2D plastic, but we're taking all the gamish. We're taking all the tumor cells, endothelial cells. In the case, as I mentioned, for glioblastoma, we have the microglia and the astrocytes and the neurons, everything in there, and we reconstruct the tumor in its 3D dimensions in a hydrogel that we synthesize in the lab, which is biocompatible and allows for it. This is a bit of a sickly idea of a chemical engineer in my lab that is doing it in the form, the Bob Langer way, in the form of an ear. But this is the tumor being here, and the reason you see it in red is because we infected the cells with M-cherry, with fluorescent protein, that we can actually follow them and see how they develop. And then we put it in our dish, but this time it's not only tumor cells on 2D plastic, but rather 3D, and now we're working on adding the microfluidics and having some flow in the blood vessels that are created in the 3D tumor that we reconstructed. This, we hope, will give us a much faster answer. We will be able to recreate and do like 100 mini-mes of that tumor and now be able to screen different molecules. This is for translational medicine, for screening reasons, but also go to the basic biology and then check how the crosstalk is going between the tumor cells and the stromal cells, and according to that, try to, as I said, interrupt those interactions. So from every tumor that we get, we do these 4 things. We take the FFPEs, those are the formalin-fixed paraffin-embedded tissues for histology. We create cell lines, and we create those 3D tumor models, and we continue with the PDX, with the patient-derived xenograft in the mouse, because we still think there is value to it, not necessarily for immunotherapies, because they don't have, these are genetically modified mice to be immunodeficient, so they do not reject the human tumor, the human tissue. And then we test on all those 4 and see, do we get a correlation between what we find in the lab and what we see in the patients. So one of the reasons to do it, besides trying to get a faster method, is actually trying to go with a personalized medicine, and we didn't invent that, not this century, that's for sure. Hippocrates already said that it is far more important to know what person the disease has than what disease the person has. And I can assure you that this morning I had to decide which kid gets which cereal, and that took me like an hour. So I'm sure if I do a mistake, that's really a problem. So this is an overview of the different models that we created in the lab. We already have for osteosarcoma, as I said, glioblastoma, breast cancer, prostate, melanoma, neuroblastoma, and ovarian cancers. We created pairs of dormant and fast-growing, metastatic versus primary, and drug-resistant versus drug-sensitive. Today I will tell you a special project by an outstanding student, PhD student, Adas Gibori, that is working on PDAC, pancreatic ductal adenocarcinoma. And we use the way to target these tumors. We use different nanocarriers. With Rainer Haag from Freie University in Berlin, we have a wonderful 10-year collaboration already, very fruitful collaboration on polyglycerol amine dendrimers, where we create polyplexes with siRNA and microRNA. These are aminated, so they are positively charged, and they create and form a tight polyplex with a negatively charged oligonucleotide. We synthesize by raft living polymerization HPMA, hydroxypropyl metacrylamide copolymer, which we bind to its several drugs or a drug and turn on probe, fluorescent probe at a near infrared wavelength. This is the probes are synthesized in Doron Shabbat's lab, and with him we also have a long-term collaboration. We synthesize in open ring polymerization PGA, polyglutamic acid. Today I will show you a polyglutamate that we modified the acid here with an amine, 50% with an amine and 50% with an alkyl. So we create this micellar nanostructure, which again, the supramolecular structure of those polymers that we synthesize in the lab has a major effect to how they will behave and what will be their pharmacokinetics in the body once we inject them intravenously. For those, we add some targeting moieties. One of them is mannose to P-selectin, and indeed it is not very selective, so I will be really happy when you'll have some more specific molecules to that. But we can see very nice targeting, or sulfate, for example, on the polyglycerol dendrimer. We synthesize by solid phase peptide synthesis an NCAM targeting peptide and alpha V beta 3 integrin targeting peptide. This is an RGD, bicyclic RGD peptidomimetic, or a folic acid, or alendronate. This is a bisphosphonate that is targeting the bone. And all this, we can combine it on those hyperbranched polymers, micellar structures, nanoparticles, or dendrimers. So now I will move on specifically to talk about the PDAC project, the pancreatic ductal adenocarcinoma, led by Adas in my group. And one of the major questions in the lab, coming back to my first slide, was if we take dormancy as a tumor that remains for a very long time at a small size in the body, can we look at patients, a very small percentile of patients with PDAC, with the ductal adenocarcinoma in the pancreas, that stay alive beyond 3 years. Those are around 3%. Only 7% survive more than a year. So it means that most of the patients are short-term survivors, and they live around 3 months. If we look back into the TCGA data, this is a bank data with a lot of patients that we specifically targeted PDAC patients. So we could find that, and this is too short time to tell you how we did it. This is in collaboration with Eitan Rupin. And we found that there is inverse correlation between the expression of Mir-34a levels and Plk1, meaning that if we have a high Plk1, which is a polo-like kinase, and low Mir-34a, this leads to long survival. If it is other way around, we see short survival, very poor prognosis. According to this algorithm, we went on and tested, we collected samples, FFPEs, as I told you in the beginning, taking slides from the patients, those that we already know that they were short-term survivors, about 3 months survived after diagnosis, or those that survived more than 3 years, which is again, are very few, about 3%. And when we found and collected all these FFPEs, we were able to stain them and to do the real-time PCR, and indeed, we found that the Mir-34a was higher in the long-term survivors, and Plk1 was lower in the long-term survivors. So this is not just an algorithm. We see that this is real in-patient clinical samples that we can collect and now validate it in the lab. So the idea was, can we combine now restore the oncosuppressor Mir-34a activity, take it up, and knock down an oncogene, in this case the Plk1, at the same time. Because when we looked at each one alone, there wasn't a significant difference between the short-term and the long-term survivors, only when we took these two candidates. Combining them together, we hypothesized that this will reverse the tumorigenic properties, improve the therapeutic response, and prolong survival. But this is a nice idea, but it's not easy to deliver RNAi. If you go to the industry, you'll see how many companies in this field had shut down their companies in the last 2 to 3 years. This is a major challenge due to aggregation in the blood, degradation by RNases in the bloodstream, engulfment by the reticuloendothelial system, the RES. There is really rapid renal clearance. Targeting to tumor site is a huge challenge. If we compare it to chemotherapy, with chemotherapy we are worried that there will be toxicity to the normal healthy organs. In this case, it's not toxic to anything. It just causes immunogenicity, but it's not arriving anywhere because it's degraded already in the bloodstream. So, and even if we get it to the tumor tissue, can we get it into the cell? This is a negatively charged oligonucleotide. We're talking about 20 nucleotides in size. This cannot, that is negatively charged, it will not penetrate and cross the cell membrane. And even if we manage to do that, it mainly get stuck in the endosome. It is not escaping from the endosome, reaching its actual cytoplasmic target, the mRNA of that protein that we want to knock down. So dealing with all these challenges, we decided that we will go, and this is why, I guess why I'm here, with a nano solution, and not because it's a small solution. In this case, it's a large solution, because usually most of you, mainly in the exact sciences, think that nano is pretty small, but for the blood vessels, this is very large. When we talk about a molecule, a macromolecule of 20 to 100 nanometers in size, like our polymers, you can see here in the tight blood vessels, these are normal, healthy blood vessels. This is by non-invasive intravital imaging. We can see that our polymer, when it's labeled with fluorescein, isothiocyanate, FITSI, it is in the blood vessels, very tight endothelium, and nothing comes out. The background is dark. But when we move 1 millimeter aside, and maybe if we can take the light, dim it a little bit, is it possible? Or can you see? And we inject it intravenously. When we move 1 millimeter aside, everything is extravasating outside due to the leaky vessels. Those leaky vessels are angiogenic vessels. They are new blood vessels that were created very rapidly with the help of VEGF, vascular endothelial growth factor, that is secreted from tumor cells, and that induces vascular hyperpermeability. So if we look at small molecules, when we inject them, they go around the bloodstream, they will diffuse out of the normal tight endothelium, but at the same time, they will also extravasate from the leaky vessels. But this is the cause for toxicity. This is why all these small molecules get to the normal healthy tissues. But when we put them on the polymer, when it is in a nano size, which is again large for our blood vessels, it will circulate for much longer time than the free entity and extravasate through these holes, through this fenestre of the endothelium, and the new endothelium, the new blood vessels in the tumor, and then be entrapped there and slowly internalized by endocytosis into the cells and release their drug load, or in this case, the siRNA and the microRNA. So as I mentioned, we synthesized by open ring polymerization NCE. We synthesized polyglutamic acid. Then we modified half of the pen groups to the acid to ethylenediamine, and half of them to an alkyl. And in this case, it was 6 carbons. We found that about 5 to 7 carbons is the best fit. More than that, it will not work, and it will not create this kind of micellar spherical structure. As you can see here, at the size of 189 nanometers in average, you can see it by the DLS and by the SEM. And also, we see that the N2P ratio, this is the ratio of nitrogen to phosphate. You can see that at the N2P ratio of 2, we have here in this electrophoretic mobility assay the neutralized polyplex of here we have a positively charged polymer with the negatively charged RNAi, and this is a combination of the siRNA and the microRNA, Mir-34a together with the Plk1 siRNA together, and polyplexed, and in this ratio we see that it remains in the well. That means that we managed to neutralize it, or close to neutral. As you can see here, about the zeta potential is about 4. And we looked at one way to see how the complex is formed and how stable it is doing a competition assay with heparin, and indeed we see that it is pretty stable and it competes with heparin later, so there is a competition with other negatively charged molecules. This polymer, what I didn't tell you, it's based on polyglutamic acid. So the amide bonds are degraded by cathepsin B. And cathepsin B is overexpressed in many tumor cells and tumor endothelial cells, and using an activity-based probe that Galia Bloom in the lab of Matt Boggio at Stanford created, we evaluated the cathepsin B activity in those pancreatic ductal adenocarcinoma tissues. And indeed, as you can see here in red, there is really high expression of cathepsin B in these tumors. That means that we can use this polymer. We have very stable polyplex with the siRNA and the Mir-34a while it is circulating. But once it will arrive to the tumor tissue and encounter with the cathepsin B that is overexpressed in the lysosome of those tumor cells and tumor endothelial cells, it will be degraded, and then we get the release of the oligonucleotide. So here we can see that it indeed internalizes. I'll go faster with this data. It internalizes into the cells via the endosomal lysosomal pathway, the endocytosis. We see an endosomal escape from the endosome to the site of action in the cytoplasma. We see that it is biodegradable, biocompatible, because it does not induce IL-6 and TNF-alpha secretion from PBMCs, and it does not cause hemolysis of red blood cells. And this is just to show you that we have done all the controls, but very quickly it inhibits, the combination is a synergistic combination of the two oligonucleotides, as you can see here, by inhibition of cell viability, cell proliferation. It inhibits, the combination of the two inhibits significantly and synergistically the clonogenicity of these cells, and it inhibits the migration of cells. That means that we will be able to inhibit the migration and the invasiveness of pancreatic cells to other organs, meaning that we will be able to inhibit metastasis. So indeed you can see the leaky vessels also in pancreatic cancer compared to normal pancreas, and we were able to inject it intravenously, so we don't even need to know where the tumor is. It finds itself, and as you can see here in red, M-cherry labeled is the cancer, and with Cy5, the blue here, it localizes in the tumor tissue and nowhere else. You can see it much clearly here. M-cherry is the tumors, and Cy5 is the actual polyplex that is labeled with the Cy5, so we can see it accumulating only in the tumor site. And last but not least, as you can see here in red, it inhibited the polyplex with a combined. All the controls in the middle are each one alone and a control non-targeting siRNA and microRNA. But in red here, you can see that we inhibited significantly tumor growth of the pancreatic adenocarcinoma in mice. This is orthotopically inoculated in the pancreas in the mice, as you can see here in red, and here you don't see it. This is the combination treatment, and it does not induce weight loss in the mice and prolongs survival. So this was not toxic, and that means that we can now increase the dose. Here we stopped it at a day after 3 weeks. It means that we are able to prolong the treatment and see if we can get indeed a longer survival without toxicity, and here you can see that all the targets were indeed silenced. One thing that was very interesting is that we saw that both Plk1 siRNA and Mir-34a inhibit MYC, and MYC is a proto-oncogene, and our model, our hypothesis is that what we are doing is synergistically inhibiting MYC, where we see also in the treated tumors, we see very nice inhibition of MYC that led to this nice inhibition of tumor growth. So I won't go through the summary, it's just what I finished saying here. This work is done with amazing 30 people in the lab. This is a very nice collaboration. Adas, as I said, did the biology, Shai and Adva and Anna help with the chemistry. We have chemical engineers and MDs and that give us mainly surgeons and oncologists and pathologists that we get the samples from. And of course, the funding. Thank you very much.
Thank you. Questions?
I have a question relating to the Mir-34a. It is known that methylation or hydroxymethylation of DNA is important for several types of cancer. And I wonder if you know something about methylation or hydroxymethylation of Mirs, and specifically about methylation or hydroxymethylation of Mir-34a.
We haven't looked at that, but it could be very interesting. We haven't looked at the methylation.
Okay.
Maybe Yuval can help us with that.
I want to take you just back to your very first slide about those micro tumors.
Mm-hmm.
So, do you see a very big difference in division rates between the vasculated and non-vasculated, for example?
No, it's actually amazing. When we look at those tumors, we see, and we got FFPEs from those in situ carcinomas, we see that they proliferate very fast. At the same time, they undergo apoptosis. So they went through the mutations. We even collaborated with Batsheva Kerem looking at DNA combing. And she did that, and she called us really disappointed, saying that she didn't see any difference between the dormant and the fast-growing. And indeed, when we grow them in vitro, they all have the same proliferation rate, same migration, invasion. All the processes that we tested in vitro are similar. Only when we inoculate them in mice or in living tissues, we see that those that grow very fast very early on can recruit blood vessels and kill the host, in this case is the mouse, within a month, while the dormant ones remain dormant as a tissue. They are proliferating and dying, but they remain dormant for a year or more, which is half the time that a mouse lives, yeah.
Okay, thank you very much.